Okay, so I know I have some videos already on my page about Introduction to Macromolecules from AP Bio. However, in the year 2025-2026 school year, College Board has made some updates, and there's just one particular word and phrasing that I don't have in my other videos, so I thought I would update it to be the most accurate and reflective of the new curriculum. Okay, so let's go ahead and talk about the introduction to organic molecules. In my previous video, we talked about carbohydrates, lipids, proteins, and nucleic acids. So these are our large organic molecules or macromolecules. So 3 out of 4 of those molecules are built in a similar fashion. So these macromolecules often begin with what we call a monomer. Now that monomer will vary depending on the macromolecule. For example, in carbohydrates, the monomer is glucose. In nucleic acids, the monomer is a nucleotide, and in proteins, the monomer is an amino acid. So depending on the macromolecule, they have different monomers, but a monomer is a single building block. To build a large molecule, we're going to take multiple monomers and join them together into a polymer. So mono means one or single, and then poly means many. So here I have many monomers that we can join together into a chain called a polymer. And so that polymer will be different for carbohydrates and different for proteins and different for DNA. However, it is all built in a similar fashion of repeating monomers joined together. So a polymer is a large molecule made of many monomers bonded together in a chain-like fashion. So we can also take a polymer and break it apart into individual monomers. For example, if I eat starch is a polymer of carbohydrates. When I have French fries, I'm eating a long chain of carbohydrates, and in my mouth I have enzymes that are going to break that large carbohydrate into individual glucose monomers, so that way I have energy for my cells. Okay, so let's go ahead though and look at some analogies for us. So a paper clip would be a monomer, and then a chain of paper clips would be a polymer, right? A single train car, and then a train, etc. So you have your monomer and you have your polymer. You'll notice in the polymer, they're not all identical, right? I on purpose picked different color paper clips or different color train cars or different color Christmas lights to show that they're all Christmas lights, but they do come maybe in different versions. Okay, so how do we do this though? How do we join small molecules together into larger ones? So if we take 2 monomers, what's going to happen is a process of removing some elements or some atoms to join the 2 monomers together. So we will remove a hydrogen from maybe monomer 1, and then a hydroxyl group or an OH from monomer 2. And if I look at that, I see H2O, so that forms water. And then as we've removed water from those 2 monomers, they come together and they form a covalent bond. So when we carry out the process of joining 2 monomers together, we remove the water, we essentially dehydrate the molecule, they've lost the water, and we built or synthesized a covalent bond. So this process is called dehydration synthesis. We've dehydrated the molecule and we've synthesized or built a covalent bond between the 2 monomers. Now we can continue adding on and continue carrying out dehydration synthesis to continue to build this long chain of repeating monomers. So if I look at my text on the screen here, dehydration synthesis is when a hydrogen ion is removed from one monomer and a hydroxyl group is removed from the other. And then a water is formed from these reactants, and then a covalent bond is formed between the 2 monomers. And now this is going to happen in building carbohydrate polymers, large protein polymers, and as well as like DNA and RNA as well. So we do carry out this process of dehydration synthesis to build. Now what happens though if I want to break this bond? Let's say I eat a large polymer and my body needs to break it with enzymes into smaller individual building blocks, into smaller monomers. What's going to be the opposite, right? We're going to take this water and we're going to put it back basically into that covalent bond, and we're going to break the covalent bond back into individual monomers. Now why I chose to make a new video on this is because this word cleaving. So College Board, if you're watching this for AP Bio, in their text to us teachers of what to teach, they use the word the cleaving of covalent bonds. And I know for my students that would probably be a new vocabulary word. So here we can read their text together. A chemical reaction involving the cleaving of covalent bonds. When water is added to the bond between the monomers in a polymer, the bond is broken. The hydrogen ion from water is added to one monomer, while the hydroxyl group is added to the other monomer. So this process of splitting or cleaving is called hydrolysis. Now in biology, when we see the word lysis, lysis means to split. So in glycolysis, we split glucose. In cell lysis, the cell membrane splits open. So here we have water, hydro, and lysis meaning to split. Now this word cleaving, you can think of like a meat cleaver that like chops meat in half, right? It's a cleaver, it's like splitting it apart. I don't know, but I made this whole video just to emphasize the word cleaving, so the first time you see it isn't on that AP test in May. All right, hopefully this was helpful, and good job.